Hello. So we are talking about chapter 9 in this video lecture, and it's a basic overview of the nervous system in terms of the brain and spinal cord. So this is a little bit of background, and you can always refer to additional videos from anatomy, and that would be chapter 14 and 16. 14 would be the spinal cord, and 16 would be the brain. Those also work. We're adding a little bit of physiology in this one, but not much. And so let's talk about blood supply. Um, so what's interesting is that not only do your neurons have a very high metabolic rate, which means they use a lot of a lot use a lot of ATP, right? And so they are consuming a lot of oxygen. Why? Because as you guys know now, oxygen is needed to create ATP. And so even though you're laying in bed and you might be reading books or studying, you're still actually using energy. A lot more than, say, if you were watching television and you're not truly having to use the neurons as actively. So that's something to think about. Brain uses about 50% of the glucose of the body at rest because the brain does not use fats for energy sources. It only uses glucose. And that's another thing to be really important, kind of cognizant of, is that your liver stores glucose for, number one, your nervous system. Has priority, right? For nervous system, number one. And that's why if you have really, really low blood glucose levels, it gets low, your brain starts to feel a little fuzzy. You can't concentrate very well. It's because those neurons are really using glucose. And if you're running low on glucose, then the neurons aren't going to have their fuel. So choroid plexus, this is where you create cerebral spinal fluid. Cerebral spinal fluid is what makes contact with the neurons. It's really turned over 3 times a day or every 8 hours. So you're constantly having these ependymal glial cells pull nutrients and fluid and ions and electrolytes and so forth out of the cerebral capillaries and create this fluid called cerebral spinal fluid, which actually is what the neurons exchange with. And so when people talk about a blood-brain barrier, they're really talking about the ability for something to come out of your cerebral blood flow and make it into the cerebral spinal fluid. So that's blood flow. Like I said, neurons only use glucose. And as it states here, only does aerobic glycolysis, and that means no beta oxidation to use triglycerides. So it's glucose dependent. There are no glycogen stores, a little bit in some glial cells, but really none for the neurons. And so that's why it's imperative that you keep a steady stream of glucose for your nervous system. And that is really the job of the liver. Skeletal muscles each store their own glycogen stores, but the neurons are relying on the glycogen stored in the liver when blood glucose levels get too low and you can't eat or drink any kind of carbohydrates or sugars in. Ketones are used during extreme conditions, but that is not something that your body can maintain for long periods of time. By the way, a stroke is caused by a decrease in blood supply. An aneurysm is where there's actually a break in the blood vessels, just to clarify. So the spinal cord, it's continuous with the brain. And your spinal cord is where the cell bodies of the neurons are located. And the axons are either coming into the spinal cord or going out, right? So if they're coming in, they're going to be afferent sensory axons, and if they're leaving, they're going to be efferent motor axons. We'll talk about the ganglions in a minute, but if you look here, there's these enlarged areas, and these are ganglions. And we'll talk about the ganglions. You have the dorsal root ganglions. Those are for sensory afferent neurons. And then you're going to have the sympathetic ganglions. That's for the sympathetic nervous system or your fight or flight autonomic nervous system. So we'll talk about that in a subsequent chapter. By the way, the axons, either coming in and going out, they're bundled together. And that is what we actually call a nerve. So a nerve is going to be axons of both sensory in axons and motor out axons bundled together. So nerves are really just axons. And what dermatomes are, so remember, nerves are mixed. So spinal nerves are mixed, meaning they're mixed both axons of afferent sensory and efferent motor. So these axons are coming in and out, and they're bundled together in what we call nerves, spinal nerves. They're coming out of the spinal cord. And since they're sensory and motor, that means they control muscles in specific areas, and they also bring in sensory information from specific areas. And so we have mapped each spinal nerve in terms of what area you have sensory receptors that are coming in. Those axons of those sensory receptors are coming in to a specific area of your spinal cord in between two vertebrae. And so if it's coming in between the skull and C1, it's cervical nerve 1. If it's C2, it's coming in above C2, below C1. C3 comes in above C3, below C2. So the spinal nerves are really named for the intervertebral foramen that they're coming out of. And so for cervical, they're named for the intervertebral foramen above that cervical vertebra. When you get to the thoracic, it's named for the intervertebral foramen below that vertebra. So T1 spinal nerve comes out below T1 vertebra, and it goes all the way down for lumbar and sacrum. So it's only cervical, it's only above, the rest of them are below. And so as you can see, we have a mapping of the area that sensory receptors are located, and those axons of those particular sensory receptors monitor that area of the skin. And so this is how you can do a nerve conduction test really easily by, stimulating a certain area of skin and seeing if the patient can, quote, feel it, because that means they have sensory receptors that are properly working, going from that area of the skin up to their brain. And you can assess which spinal nerves are working and which spinal nerves may not be working. And you assume that if the sensory is working, that the motor is also working, provided they don't have any, diseases like multiple sclerosis or something. So gray matter, that is the cell bodies. So gray matter are cell bodies of neurons, and the white matter are axons. So gray matter, you have the horns, right? So you have, it's shaped kind of like an H here. And so this is the gray matter, right? And this is the white out here. And so these are the axons, and here are the cell bodies. And so it's showing you a cell body right there. And it's always really, truly motor in front sensory, oops, in back, right? So remember this, motor in the front, sensory in the back. And so when I say that, what I'm really saying to you is that the ventral horns are going to be motor neuron cell bodies. And the dorsal horns are going to be sensory neuron cell bodies, because it's motor in the front, sensory in the back. So the white matter tracks that are predominantly all in the front are going to be motor tracks, meaning the axons are going down, they're descending tracks, and they're going out to skeletal muscles and glands, or cardiac and smooth muscles. So anterior tracks, descending tracks, those are motor tracks. So descending, anterior tracks, motor tracks. And then, you know, these ascending posterior tracks tend to be sensory tracks going up, and these are going down. The spinal cord. And so kind of the setup is that you have sensory in the back. So dorsal root, that's going to be a sensory root, and the dorsal root ganglion are going to be the cell bodies of the afferent sensory neurons, because it's sensory in the back, so they're coming in. And then the motor, that's a motor neuron cell body, is going to be located in inside the ventral horns, because that is it's just a multipolar neuron. So the cell body is at one end and the synaptic terminals are another. With sensory, as you can see, you have sensory receptors, long axon, cell body, another axon, and then the axon terminals are going to synapse then inside the dorsal horns on a second sensory neuron that will go up. The axon will go up the ascending tracks. So understand ventral and dorsal as it pertains to motor and sensory, and the axons and the cell bodies are in the gray matter and the tracks are in the white matter. So that's kind of what this is going through. So this is showing you the white matter tracks, again, ascending tracks, those are going to be sensory, right? They're coming in. So sensory tracks are bringing sensory information into your brain. Now, as you can see, there are some sensory tracks that are going to be in the front, and those are going to be specific for motor. So spinothalamic tracks and spinal tactile tracks, these are ones that are dealing with a lot of times posture, balance, and coordination. As you get more to the lateral side and dorsal side, they become more pure sensory. The descending tracks are motor, and they're coming down and they're going to be controlling your skeletal muscle. To the extent of what you need to really understand, right? You need to understand this at a basic level. Pathway for ascending and descending tracks. So we will talk about this in the next lectures, but you know, with sensory ascending tracks, sensory ascending tracks, there are really 3 neurons that make up that track. You know, basically a sensory input to brain. So the first one is right here. It's from the receptor And here's your dorsal root ganglia. That's the cell body. And then it will synapse here on a second sensory neuron, right? Ascending. I shouldn't say CNS because it's in the CNS, in the brain. And first thing I want you to realize is one, here's where it crosses over, and this is called desiccation. So you might prick a thorn with your left finger, and so you have a pain receptor that will become stimulated on your left finger. It's going to come in at the level of the spinal cord, somewhere in your probably lower cervical area, upper thoracic, one of those spinal nerves. It's going to synapse on a second sensory neuron in that area, dorsal horn, gray area. And it's going to cross over. That second sensory neuron's crossing over to the opposite side of your spinal cord and going up the opposite side, so the right side. It will go on a right ascending track, and it will synapse in the thalamus on the right side, right? So left receptor, but it synapses on the right thalamic hemisphere, and then will synapse on a third sensory neuron here in the thalamus. And that third sensory neuron will then end up in the somatosensory cortex, or primary sensory cortex, on the right. So what I'm trying to say is the sensory information that is being collected on the left side of your body is being received in the right hemisphere of your brain. And it's a 3 neuron track. So you have a first sensory neuron, a second sensory neuron that just goes up the spinal cord to the thalamus. Everything synapses in the thalamus, all sensory information, except smell, synapses in the thalamus and then goes to its cortex area. And if it's general senses, it's going to the somatosensory cortex and then a sensory association area. Motor, on the other hand, is, the first neuron, and there's really only 2. There's an upper motor neuron and a lower motor neuron. And so instead of 3 neurons, it's only 2 neurons. And my writing's getting really bad. But so the first neuron is going to originate in the primary motor cortex. So upper neuron, primary motor cortex. And it's going to go all the way down. Now, with these pyramidal tracks, these are your tracks that are basically controlling most of your skeletal muscles. I should say all your skeletal muscles to an extent. This upper motor neuron does cross over your spinal cord in the medulla oblongata, the very end of your brain, and then continues on down the opposite side of your spinal cord. So it's showing you that this motor command originates on the right primary motor cortex. That's an O. And then in the medulla oblongata, it's going to cross over and then synapse here on the lower motor neuron and go out to effect. Remember, these are effectors. The left side skeletal muscle. And so these are really long. These motor neurons are really long. There's only one synapse at the level of where that muscle is located, in that spinal cord or spinal nerve. And this is really important, especially when you're assessing stroke patients. So if you ever work in an ER or EMT or whatever, and you're assessing a stroke patient, what is important to remember is that when you ask someone to lift up both of their arms to see if they've had a stroke, and their left arm lifts all the way up to shoulder height, but their right arm doesn't, that's going to tell you that the stroke is in the frontal lobe of the right hemisphere. Wait, and I forgot if I said, so let me write this down. So, because I was like, did I say right? Did I say left? Okay, so stroke, right? Someone have a stroke. So if you were to ask lift both arms and right arm comes up to shoulder, but left arm does not. You're asking them to control, a skeletal muscle. And remember, it's originating in the primary motor cortex of the frontal lobe. And again, I cover this in chapter 16 of the anatomy lecture. So if you really want to get into it, you really want to know this, you can review, the anatomy part of this. But if you have a stroke and you're trying to assess which side, the right arm seems to be working fine, but the left arm doesn't, that's really telling you that it's a left hemispheric stroke, right? So that's not a very good color. So that's basically saying stroke was in left, oh, sorry, I'm not paying attention, was in right frontal lobe, so right hemisphere. Because of this crossover. So your left primary motor cortex controls the right side of your body, and the right primary motor cortex controls the left side of the body. And your sensory receptors on the left side of the body are going to end up on the right hemisphere primary sensory cortex, or your postcentral gyrus, and be interpreted first on the left side, and vice versa. So you have this crossover. All right, so I'm just going to kind of review the anatomy of the brain. You have a forebrain, a cerebellum, and a brainstem. So the forebrain is the cerebrum, it's your cerebral cortex, and the basal nuclei for motor. And this is where really all the neurons that are responsible for thinking, right? Thinking and controlled movements, etc. Personality, whatever. This is like cerebral cortex. Basal nuclei is for smooth muscle movement coordination. And so that's number one. Number two is diencephalon. It's kind of your subconscious brain. You have two thalamic nuclei, one on the right and one on the left. These are a collection of cell bodies. It's not the nucleus of a cell, it's a nuclei in the brain, which is really a grouping of cell bodies. So you have the thalamus on each side, and then you have one hypothalamus, and we'll get into all the things the hypothalamus does. It's quite extensive. And then you have the cerebellum. It's directly across from the pons, which is part of the midbrain. So you have midbrain, pons, cerebellum, and as you can see here, right, they're directly across from one another. And then you have the medulla oblongata here that is continuous with the spinal cord. If you guys watch my video, you got your seahorse right here. So seahorse is diencephalon, midbrain, pons, and medulla oblongata. So this goes to the cerebrum. Diencephalon, it's showing you the thalamus. Now here's one thalamic nuclei, and it's in 2D, but it's a roundish structure in 3D, and it's only showing you one side. So you're only seeing one thalamic nuclei. This little spot right here is where the two thalamic nuclei touch. And so that's called the thalamic adhesion molecule, and the third ventricle for cerebral spinal fluid is located, it's a space between the two thalamic nuclei. So that's what you're looking at there. The hypothalamus is the space here, and this would be the pituitary gland. It's attached to the hypothalamus. Hypothalamus is part nervous system, part endocrine. So go over that in chapter 19 of anatomy, right? So if you want to know about the hypothalamus and pituitary, it's chapter 19 of anatomy. Midbrain. Midbrain has some important functions. It's really this right here. You have little nuclei for reflexes for vision and hearing, and you have the reticular activating formation that keeps you kind of alert and on standby. You have a lot of tracks going through there, a lot crossing over. The motor tracks there. You have some nuclei that are producing neurotransmitters that are involved in smooth, wanted motor movements. So there's stuff going on there. And then you have the pons. Pons has some centers for breathing and respiratory cardiovascular centers. It also is kind of a relay for sensory information that's proprioception for balance and equilibrium, and muscle spindles and Golgi tendon apparati, things that are telling your brain what your muscles and tendons and joints are up to. It's going to synapse in the pons on a neuron that then goes over to the cerebellum. So it acts as a relay for cerebellum to receive proprioception information. And then the medulla oblongata has some really basic functional respiratory, digestive, and cardiovascular centers, and is continuous with the spinal cord. So you have a lot of tracks there. The corpus callosum is axons, neurons that communicate between hemispheres, right? So you have sensory information coming in your right hemisphere, and it wants to share it with the left hemisphere. You have neurons that cross hemispheres. These are commissure neurons, and they help the right and left hemisphere communicate with one another. And the axons are of these neurons that communicate between the hemispheres are all located in the corpus callosum and also the anterior commissure, which is up here. So I'm just explaining to you what these are. I'm not necessarily testing you on the anatomy. I'm just kind of going through the process of explaining. Cerebellum is your motor brain. We'll talk a little bit more about that. And medulla oblongata. Here's kind of the short list from the anatomy book. Review with the cerebellum and the medulla oblongata and pons, mesencephalon, which is the same as midbrain, and diencephalon and cerebrum. So it just kind of goes through the basics of what each does. Cranial nerves, just touch on this. You have 12 paired cranial nerves. They're called cranial nerves because these nerves originate on the brain or brainstem. And there's one on the right, one on the left, just like spinal nerves. But unlike spinal nerves, which originate from the spinal cord, these nerves can be pure sensory in or pure motor out, or they can be mixed. And so some of these are really confusing to me in this table because certain books will classify the ocular motor and trochlear and abducens as pure motor. And I'm not sure why your book, it's very tad. I mean, when you're dealing with balance and equilibrium, yes, there is some proprioception, but it's that this nerve is working with that system. And so don't get confused when like the anatomy book will term the abducens as pure motor, controlling the eye muscles. And for some reason, this book has them as mixed, but you don't need to worry about that. I just want to bring up the point that cranial nerves can just be afferent sensory axons coming into the brain, or it could be pure motor axons leaving the brain. And that's what that nerve class means. But they're all dedicated to different sensory and muscles, usually at the upper portion of your body, except for the vagus nerve, and that is your parasympathetic nerve, right? So cranial nerve 10 is the vagus nerve, and when we talk about the autonomic nervous system, which is sympathetic and parasympathetic, the vagus nerve is really the primary nerve of the parasympathetic nervous system. It's a cranial nerve. Lobes, right? You have frontal. Let me change the font there. Frontal. Well, that's not even a very good font, is it? Let's come up with something a little brighter here. Frontal, parietal, temporal, occipital, and I know the names are there. And this is cerebellum. And it's kind of showing you the sulcus that kind of differentiate the lobes. Each lobe, this will go through central sulcus is kind of the boundary, you know, from the frontal lobe and parietal lobe. And again, when I say it's motor in the front, sensory in the back, this is the primary, let's go back to this, primary motor cortex. And this is the primary sensory cortex. And remember, I'll say it again and again and again, motor in front, sensory in back. And so what you're going to find is, that in front of the central sulcus, these are motor neurons. So that's primary motor cortex and premotor cortex. And then you have the prefrontal cortex, and that's a little bit different. This is really intellectual thought, personality, you know, deep thinking here. And this prefrontal cortex is the last to be to develop. So your brain starts to develop from the basically the brain. It starts off a hollow neural tube, and that kind of is your spinal cord, and it morphs up into a brainstem, and then it morphs up into a diencephalon and cerebrum, and so it develops really from the bottom up. And so your brain's developing basically in this pattern. It's going like this, right? And so you're developing, and the last to be developed is your prefrontal cortex in your 20s, right? So it can even be up to 26. So this is where real decisions are made in your prefrontal cortex. This is where you can extrapolate experiences and predict what is going to happen. Here's where you make these complex kind of abstract judgments on what to do. And this is not developed into your 20s. And so at 16, 17, 18, you know, when your parents asks you, why did you do that? Right? You did something really dumb that any 40-year-old could have predicted was going to happen, right? But to a 16-year-old, they can't predict that that's going to happen. So they do dumb things. And of course, what everyone knows is going to happen happens. And so it's the same bad outcome. And your parents are mad at you and they're like, why did you do that? And you're like, I don't know. The fact of the matter is, if you're 16, 17, 18, you probably don't know. You don't have a functional prefrontal cortex. You're not able to really sit there and go, okay, let me think about what's going to happen in the future. And so there's, you know, this really is a critical thing to think about when you're talking about education and decision making and what are you going to do with the rest of your life. Well, if you're 18, how do you know what you're going to do with the rest of your life? Your prefrontal cortex isn't fully formed, right? It's really hard for most humans to make these really abstract, concrete decisions of a 60-year working career based on the fact that they don't have critical parts of the brain that can think that deeply. So I think it's important to kind of understand the way the brain develops. It's also why teenagers tend to have more risky behaviors than adults, right? So adults are going to be like, oh no, I'm not going to do that because it's, you know, here's what's going to happen. But teenagers are like, hey, what possibly can happen? Or they just don't even think about it at all. prefrontal cortex. Developed, not developed. So prefrontal cortex and motor is the frontal lobe. Behind the central sulcus is all going to be sensory input, right? So we're looking at this, and this is all, sensory input. And what is the good word? Processing, right? A couple of things. So we'll go through the primary somatosensory cortex. This is where all general sensory input ends up after thalamus. And so when I say general sensory, it's everything except for vision, hearing, smell, taste, and balance and equilibrium. And so that includes everything else. That's the primary somatosensory cortex. So that's where it goes after synapse and thalamus. Then there's the sensory association area. So you might have pain receptors that light up, and you also have vision information coming in, you have auditory information coming in, you might have temperature and all kinds of other things. So all these receptors are lighting up at the same time. If they're special senses, they're going to go to their special cortexes. So if it's vision, it's going here to the occipital lobe. If it's pain, it's coming here to the somatosensory cortex. But it all has to integrate. And so here's where you have integration of sensory, right? So you might have touch and pain at the same time, and temperature, right? You touch something that burns you. You have pain receptors go off. You have thermal or temperature receptors go off. And those two, they need to integrate. And they also need to integrate with what you're seeing, so you can then log it away that you don't want to do that again. And so you have these integration areas where sensory information is getting coordinated and integrated. As you get to the occipital lobe, right, you have the vision and primary visual cortex, visual association area. You have Wernicke's. This is for understanding language, right? And again, Wernicke's is in the back. It's behind, right? So here's the central sulcus. And it's posterior, so it's understanding. It's hearing language, which is sensory. It's seeing written language, which is sensory. It's feeling braille, it's sensory. So we comprehend language based on sensory input of some sort. And that's going to be different than Brokaw's area. Brokaw's area is here, and we'll get to that in a minute. So auditory is for hearing, right? So primary auditory cortex, and then the association area. And limbic system is in the temporal lobe, but it's also in the diencephalon and in areas of the cerebrum. So it's not just one area. Olfactory is smell, there, top of the temporal. And then you have Brokaw's, and this is for speech. Now, this is in the front, speech formation. And this, my friends, is all muscle. Right? Muscles control your ability to speak. And so, again, muscles are motor, and so they're going to be in the frontal lobe. So that's Brokaw's. Prefrontal cortex, kind of went through that, right? Personality and so forth. Premotor cortex, voluntary movements, and primary motor cortex. So that's in the front. The homunculus here, it's kind of showing you how many neurons are dedicated to these body parts. So it's really demonstrating number of neurons that control or monitor area of body. And so the bigger, right, so face, there are a lot of sensory receptors in the face. Why? Because the face is giant on this depiction, and it's equating size of that body part area to how many neurons are dedicated to that. And hand, obviously we have a lot of sensory receptors in our face and hand. not so many on our elbow or knee. And this comes into play when we're trying to locate a stimulus. We can very acutely locate a stimulus on our face or hands because we have so many sensory receptors monitoring little tiny areas. And so it has a high acuity. Whereas if it's one sensory receptor monitoring a huge area, we just kind of know it's there in the general area. As far as motor, we have a lot of muscles dedicated to our face and hands. They are small motor units. There's a lot of motor neurons that control just a very few muscle fibers. And so that's what it shows there. Somatosensory association area, cortex, I kind of went through this, right? It integrates sensory input from primary somatosensory. So you can kind of do touch with texture, with vision, with how it felt, with pain, with smell, all kinds of stuff. And so it's receiving these multimodal association areas, receive multiple sensory information from different parts of your cerebrum. And so you can store what sensory information is coming in, right? So you don't have to keep kind of making the same mistakes twice. So as a little kid, when you're born, your brain is like a blank computer, and you're just programming that computer from the moment you're born. And that's really just sensory input, right? And you're storing all the sensory data that's coming in. And so you can always compare new stuff to old stuff, right? A cat is fluffy and soft. And so as a kid, you learn this, but you also learn a cat whose hair is kind of sticking up and whose ears are back is going to be a different experience than a cat whose tail's wagging and it's kind of sauntering up to you, right? They're both fluffy and soft, but there's also different cues that you learn that get logged away in these multimodal association areas so you can make better decisions. So this kind of outlines the brain. Lateralization, why there's crossover, not really known why it evolved that way, but this kind of goes over what I talked about with this crossover of the pathways. There is brain specialization, brain hemisphere specialization, right? So right hemisphere tends to have more creativity, spatial perception, and the left hemisphere has more like logic, analytical language. It's not a hard and fast rule for everyone. Not a lot of people want their brain studied while they're still alive and using it. So as technology gets better, we're able to study brains a little bit better, but there's still kind of a lot of guesswork. Thalamus, okay, so we're in the diencephalon. The thalamus, again, is a collection of cell bodies of various neurons, right? And so it's kind of breaking it down into areas. So you're going to have, cell bodies that are part of the emotional limbic system, part of your brain, frontal lobe, parietal lobe, association area lobes, visual, and so forth. Remember, you have 2 thalamic nuclei. All sensory information will synapse in the thalamus. And so that's why it gets this relay center. For motor, it's involved in complex motor loops between the cerebellum and the pre and primary motor cortex. And so that's why motor pathways is also involved. It's the final relay point for all ascending sensory information before it goes to its cortex. And so that's kind of important to understand. Hypothalamus is really part endocrine and part nervous. You have some subconscious control. It's part of the limbic system, right? So this is really limbic system. And the limbic system is its whole coordination of different parts, but those parts are all dealing with emotion, pleasure, pain, rage, fear, so wide range of emotions. In addition, in this small area, because it's a very small area of the brain, You also have control over autonomic centers. So fight or flight or rest and digest of heart rate, blood pressure, respiration, and digestion. So when we get super stressed out, the hypothalamus, which is part of our emotional brain, our limbic system, you're getting stressed out in that same area. You also have autonomic control over heart rate, blood pressure, respiration, and digestion. So you're stressed out, your heart rate goes up, your blood pressure goes up, your respiratory rate goes up, and your stomach doesn't feel great, right? Because you have digestion issues. So these are all happening in a small area. Also, it's got that antidiuretic hormone oxytocin. It also controls the output of the pituitary gland, which is a major endocrine gland. It controls the adrenal medulla for epinephrine, norepinephrine, right? So that's your fight or flight neurotransmitters, really. Secretion of hormones, behavioral drives. You have your thirst center and your feeding center. So these are centers that tell your brain, oh, I'm hungry, or, oh God, water sounds really good to me right now. That's because you have these feeding centers and thirst centers in your hypothalamus that tell you that you need to eat or drink, right? It's a behavioral drive. And sometimes these get a little out of whack, especially feeding centers. We're finding that large amounts of adipocytes that produce a hormone called leptin, it stops working in the thalamus in the feeding center. And so you constantly have the behavioral drive to eat. You feel hungry, even though you have more than enough triglycerides stored on board. So obesity syndrome is really, it's not just type 2 diabetes where there's a breakdown now of the hormone receptor complex, but also we're finding in the hypothalamus with the feeding center, there's a breakdown as well. We'll talk about that a little towards the end of the semester. There's also, so autonomic functions, there's also regulation of body temperature and circadian rhythms, right? So when you wake up and when you go to bed. These are all in the hypothalamus, in addition to it being part of your limbic system, which is your emotional brain, and your learning and behavior. So it's not a mystery why when you're super stressed out, your limbic system is dealing with the emotions of stress, and that's why it's very hard to learn when you're stressed out. It's also why it's hard to control your behaviors when you're stressed out, right? So you can have outbursts, you can cry easier, you have trouble focusing, you don't remember what you're trying to focus on, so that's learning. And this is psychologists love the limbic system, and you get to learn about that in psychology, and I'm not going to get too into it right now, but those are showing you all the parts. Amygdalas for fear and rage. Olfaction, smell is a big part of your emotional limbic system. Mammillary bodies are reflexes for eating, chewing, that kind of thing. So we like to chew when we're stressed. Hippocampus is for memory. So they're all connected, and that's the limbic system. Reticular formation, this is kind of your standby for your brain. It keeps you on alert. So you have input from vision, you have input coming up, and it starts in your midbrain, and it keeps your cerebrum on standby. So you might not be thinking per se or focused, but you're not unconscious or asleep. And so this reticular activating system, it's really sending a continuous stream of impulses to the cerebral cortex, keeping you conscious and alert, but you might not be actually really using your cerebrum. It tends to filter out weak, repetitive, familiar input, right? So you're not having to process every single sensory information, kind of like the white noise, right? So you can always, if you really listen, there's usually some white noise somewhere, but you're able to filter it out, this reticular activating system. When you go to sleep, it becomes inhibited or goes to sleep. So your brain, cerebrum can stop being active. Also, alcohol and certain drugs like Ativan, Xanax, the diazepines, morphine, heroin, those kind of things, they all inhibit the reticular activating system. And so that's why people will pass out. So when you successfully inhibit your reticular activating system, you basically pass out or go to sleep. That's also why you can't remember, right? You become unconscious. If there's severe injury and the brain needs to shut down, the reticular activating system shuts down. That's kind of what we term a coma. Basal nuclei is a group of structures, cell bodies that are dedicated to movements. And so basically they control unwanted movements or unpurposeful movements from purposeful ones. And so these are really in kind of the diencephalon or inner area of your cerebrum. They're termed the basal nuclei. So the caudate, the putnam, the globus. These, you don't need to learn these per se, but I'm going to explain to you, like Parkinson's, there's a breakdown. And so in the basal nuclei, the nuclei the red nucleus that produce a neurotransmitter that works with the basal nuclei. And so in the midbrain with Parkinson's, you stop having this neurotransmitter being produced in the midbrain, the red nucleus. And so that neurotransmitter is responsible for inhibiting unwanted movements in the basal nuclei. And so you have tremors or unwanted movements happening. So if you go further into medicine, you can learn about this. Reflexes, you are born with some reflexes. Those are innate. You're born with them, genetically determined. But we acquire a lot of reflexes. Driving is an acquired reflex. Or when you see something coming at you, you acquire the reflex to duck, right? Because everyone's been hit in the face by a ball before. Well, not so much anymore because they don't let children throw balls at each other anymore. But back in the day when you can, on recess, and there were rubber balls, there was always a ball flying through the air. If you're on the field, you were playing dodgeball or whatever. And if you weren't looking, you could get hit in the face with a rubber ball. And so it happens. You then learn to duck really quick, right? We have acquired reflexes. Playing an instrument can be an acquired reflex. Certain skills in a sport can be acquired. So you learn these, right? And they can be somatic. So somatic is skeletal muscle. Visceral is going to be like vomiting, or certain smells can cause vomiting, or your heart rate can go up if you see something stressful, right? So we acquire visceral reflexes. They could be cranial or spinal, meaning they come from cranial nerves, or they're initiated or controlled by cranial nerves or spinal nerves. And they can be very simple, one synapse, or they can have a lot of synapses. And so reflexes, the definition is a rapid involuntary response to a stimulus, right? So one stimulus, that same stimulus will cause the same motor response every time. And obviously they're really to help automate stuff in our brain, but also to keep us safe. So you have different reflexes. The cross extensor is a well-known reflex. Works for arms and legs. If one arm flexes quickly, the other arm's going to extend quickly, or leg, right? So this is generally to keep us safe. It's showing you someone grabbing your arm. You're going to flex it up, and you're going to extend the other arm because you don't want to be attacked. For feet, if you step on a tack or nail, you're going to flex the foot that's on the nail, and you're going to extend the leg to support you so you don't fall down to the ground. So that's another cross extensor. We test spinal reflexes to see that your spinal tracts are working, that your nerves are working, spinal nerves. And so this kind of goes through, know the definition of a reflex, why we test for them. The components of a reflex, I believe you cover this in lab, but you have a receptor and it excites a sensory neuron that goes in. There's usually integration, so interneurons in the brain or spinal cord, and that will excite the exact same motor neurons and the exact same effectors every time. So it's like a set loop. And so this just goes through stimulus, afferent neuron, integration center, efferent neuron, motor response of an effector. And so that's a reflex. We have proprioceptors in our muscles and in our tendons. These are muscle spindles and Golgi tendon organs that tell your brain constantly what the length of your muscles are and the tension in your tendons and your joint angles. And so these receptors are constantly giving feedback to your cerebellum and your somatosensory cortex. And so you're integrating your body position all the time, even though you're not aware of it. So stretch reflex, you can go through this. I have a definition. I don't get too into it, but you should read through the stretch reflex, right? And you can go through this. And I hope you do it in lab. When it says it's going through unwanted, inhibition, right? You have inhibition, I should say, inhibition of the hamstring at the same time. Here it is. That you are contracting your quadricep muscles. So remember, we have inhibitory graded potentials and we have excitatory. So the hamstrings, it's an example in this stretch reflex that you have inhibitory graded potentials of the hamstring while you have excitatory of the quadriceps. And here's the cross extensor for the legs. So again, a cranial reflex, just an example, is like when someone shines a pen light, like a light into your eyeball, they're seeing if your ocular motor nerve is working correctly, your cranial nerve 3. It's an autonomic reflex. Your pupil should constrict if bright light is shined into it, and it will dilate if it's not, right? So they can tell if you've had some kind of with concussions, if it's bad enough and the brain is swelling, it's going to affect your pupillary light reflex. If you're on drugs, certain stimulant drugs like meth or cocaine will also affect this pupillary light reflex, right? So photoreceptors and goes into the reflex. This gives you some cranial reflexes. Motor behavior, just real quick on motor action. If you just, look, your skeletal muscles are voluntary. You decide to move them for the most part. Not postural. We're not talking about muscle tone and postural. We're talking about like actually executing some kind of movement. And you make a plan. And so there's feedback between the cerebellum, thalamus, and the basal nuclei. And so this goes through the motor pathways. And I think I kind of covered these pathways. I want you to read through this and let me know if you have questions. Okay, so this goes through the upper motor neuron, lower motor neuron, crosses over in the medulla, and the different tracks. So pyramidal tracks are for the somatic nervous system. So that's skeletal muscle, right? And then the extrapyramidal. These are voluntary. These are the tracks that you decide. If you're going into postural and muscle tone, then you get into the rubrospinal and the reticulospinal and the vestibulospinal. And I'm not getting really too into the tracks, but you need to read these slides. And I'm not going to redo the slides. It's very straightforward, right? And so these tracks all control your posture. And you have these 5 input, right? You have skin receptors, eyes, ears, right, for balance and equilibrium. So that's the vestibular complex. Proprioceptors from your skeletal muscles, tendons, and joints, and the vestibular apparatus, which is ears. So I don't know why it has that twice. And so motor coordination, a lot of times you can see you have sensory information, right? And it's coming into the spinal cord. And the spinal cord is then sending information to the cerebellum. The cerebellum talks to the brainstem. The cerebellum also then will synapse its motor plan. So complex, this is where you get really complex motor plans. And they get sent to the thalamus. Then it gets sent to the cortex, right? The primary motor cortex. And then it gets sent out to the muscles. It goes through this loop here for unwanted versus wanted movements. And so they're all kind of tied together. Now, do you have to remember all this? No, but you need to understand that all these are kind of involved in motor coordination. So especially if you're going into exercise science, right? You should get into this, maybe biomechanics, or I'm not sure what class they will address this, but here's this feedback loop for voluntary motor control, right? So you select movements and how you learn repetitive movements. So these are like skilled movements. This is where you see also that effect in Huntington and Parkinson's disease. Last thing is learning and memory. So learning is the acquisition of new information. Memory is the retention of that. And retention, we mean neural connections, right? And loops. And so you have to form these strong neural connections and loops in order to, quote, memorize something or remember it. The term plasticity is really, the definition is having a permanent change in the neural pathways or synapses, right? And so this is where you get into the debate of cannabinoids and marijuana. So, everyone loves marijuana, and it's like this fantastic drug that everyone thinks is great. Well, the problem is that the cannabinoids in marijuana basically inhibit neural pathways and synapsing, right? So you have no synapsing. And if you have no synapsing, you have no memory, right? And you have no learning. And so that is just a physiological issue that everyone is going to have to deal with at some point in society. But that is not part of this lecture. So hippocampus is where we have memory and learning coordinated. Associative learning is where you attach stimuli to kind of known stimuli. You're making associations. Generally, a new stimuli is attached to a already known stimuli. Non-associative is just you repeat something over and over and over and over and over again. You just form a new neural circuit, right? Then you have procedural memory and declarative memory. So this is learning, by the way. So attach new things to existing things, or you just repeat it over and over again and create a new loop with non-associated. That's learning. Memory, right? So recalling it is different. You have procedural memory and declarative memory. So procedural memory is implicit, right? It's understood or unspoken. So motor skills and behaviors, automatic responses, these are procedural. Driving a car, this is procedural. Your response to very specific everyday things, procedural. Declarative is more school type explicit memory, where you have to recall facts, events, languages, memorizing things. And this is where the hippocampus is involved in declarative, right? So school type memory. Oh, I have it written down, school type learning. Whereas procedural memory is more motor, learned, cerebellum is generally involved. And so there are different types of memory. Motivation, I have motivation here, pleasure. If you have this in display mode, it will link you to these podcasts. And they're just interesting, related to the brain type podcasts. And this kind of also then highlights, multiple sclerosis, Alzheimer's, Parkinson's. And that concludes this. Please write down any questions you have, and we will move on to chapter 11. So thanks for listening.